Good afternoon, everyone. Happy Christmas Eve. Thank you for joining us. Welcome back. I'm Steve.
And I'm Scott.
Together, we're Backyard Musings, the Science and Technology channel.
And we're talking about a health issue this afternoon, another health issue. So here we go.
Yeah, and just as a reminder, Merry Christmas. We're going to have a special tomorrow for you. So that's when we start our one a day until New Year's Day, and then we'll go back to the two a day. All right.
Yes, we will.
Here we go. Attention disorders such as ADHD arise when the brain struggles to separate important signals from irrelevant noise. At any moment, the brain is flooded with information, and staying focused depends on filtering out distractions while responding to what matters most. Most current treatments address this problem by stimulating brain circuits involved in attention, particularly in the prefrontal cortex. These medications work by increasing neural activity. Would you like to know more?
New research points to a very different strategy. Instead of boosting activity, the study suggests that reducing background brain activity may help sharpen focus by lowering mental noise. In a study published December 22nd, 2025 in Nature Neuroscience, scientists report that a gene called Homer1,
You made that up.
no reference to the Simpsons, I'm sure, plays a key role in regulating attention through this quieter approach. Mice with lower levels of two specific forms of the gene showed calmer brain activity and performed better on attention tasks. Oh boy.
The findings point toward a potential new direction for treating attention disorders. By calming neural activity rather than amplifying it, future therapies could offer an alternative way to improve focus. Wow, it doesn't take a genius. Why didn't I think of that? Right? The research may also have broader relevance since Homer 1 has already been linked to conditions involving early sensory processing differences, including autism and schizophrenia.
Quote, the gene we found has a striking effect on attention, and is relevant to humans, says Priya Registropathy. Whatever. Whatever. That's I don't know. Head of but whoever that is the head of the Scholar Horbach Family Laboratory of Neural Dynamics and Cognition at Rockefeller. Okay. When the research team began exploring the genetics behind attention, Homer 1 was not their expected target. The gene is well known for its role in neurotransmission, and many interacting proteins of Homer 1 have appeared in human genetic studies of attention disorders, but Homer 1 itself had not previously been singled out as a major driver of attention.
To investigate this further, the researchers analyzed the genomes of nearly 200 mice bred from 8 different parental lines. Oh, wow. That's cool. Okay, they got some diversity. So I'm sure that the mice enjoyed that, right?
I don't know.
Some of these mice had wild ancestry, a design meant to mirror the genetic diversity seen in humans. This wide range of variation allowed the team to detect genetic influences that are often missed in more uniform laboratory populations.
This broad genetic scan revealed a striking pattern. Mice that performed best on attention tasks had much lower levels of Homer 1 in the prefrontal cortex, the brain region most closely tied to attention. The gene was located in a stretch of DNA that explained nearly 20% of the differences in attention observed among the mice. Maybe that's why I don't have much of attention. Maybe I don't have a prefrontal cortex.
Maybe. Yeah. Was it ripped out, or you just weren't born with one?
I think I just didn't have one when I was.
Okay, when you were hatched? Okay.
Yeah, but I got a lot of room in there for other things, so because it's not there.
Yeah. Quote, okay. That's a huge effect, Raja Sethupathi says. Even accounting for any overestimation here of the size of this effect, which can happen for many reasons, that's a remarkable number. Most of the time, you're lucky if you find a gene that affects even 1% of a trait.
Further experiments showed that not all forms of Homer 1 were involved. 2 specific versions, Homer 1A and Anaya 3, stood out. Okay. I wonder if I'm not even going to go there. Mice with naturally lower levels of these versions in the prefrontal cortex consistently performed better on attention tests.
When researchers experimentally reduced these gene variants in adolescent mice during a brief developmental window, the results were dramatic. The animals became faster, more accurate, and less easily distracted across several behavioral measures, making the same genetic change in adult mice. However, produced no improvement, suggesting that Homer 1 shapes attention during a critical period early in life.
The most unexpected findings emerged when the team looked at how Homer 1 affects brain cells. Lowering Homer 1 levels caused neurons in the prefrontal cortex to increase their number of GABA receptors, the molecular brakes of the nervous system. This change reduced unnecessary background activity while preserving strong, focused bursts of activity when the meaningful cues appeared. Instead of firing consistently, neurons save their activity for the right moments. Smart. Leading to more precise and reliable responses. Okay.
For Gershon, the results felt intuitive. He lives with ADHD himself and says the project was personally meaningful. It's part of my story, he says, and one of the inspirations for me wanting to apply genetic mapping to attention.
He was also the first to notice that reducing Homer 1 improved focus by limiting distractions. In his view, the finding aligns with everyday experiences. Quote, deep breathing, mindfulness, meditation, calming the nervous system, people consistently reporting better focus following these activities, he says.
Cool. I mean, we could all stand to do that. Yeah. Most existing treatments for attention disorders work by increasing excitatory signals in the prefrontal cortex using stimulant medications. The new findings suggest another possibility: medications designed to calm neural activity instead.
Because Homer 1 and its interacting proteins have been linked to ADHD, schizophrenia, and autism, further research could reshape how scientists think about multiple neurodevelopmental conditions.
Give me your money. That's what they're saying. Looking ahead, Raja Sethupathy's lab plans to continue exploring the genetic foundations of attention with the goal of developing therapies that precisely adjust Homer 1 levels.
You know, I'm pretty sure he and myself like the way you pronounce his name a lot better than I do. So, okay. This is the last one, right? I'm on the last one.
Yeah.
There's a splice site in Homer 1 that can be pharmacologically targeted, which may be an ideal way to help dial the knob on brain signal to noise levels. Raja Sethupathy says, thank you. Bless you. This offers a tangible path towards creating a medication that has a similar quieting effect as meditation. Okay.
Don't you love it how we have fun? We have so much fun on this channel. It's pretty cool.
We do.
We're cousins.
It may not seem like it, but we do. And yes, we are cousins. Believe it
or not. Folks, thanks for joining us. And Merry Christmas. Hope you've been nice rather than naughty so you can get something from Santa.
We hope you have a joyous day tomorrow with your family and friends or whatever you're doing. So take care.
See you.
Thank you.